know their names. Really?
Okay, I think we should start again. This is our last restoration seminar, and before we do anything, I just would like to announce for the students that they must submit their summaries or whatever they still owe me, because next week will be the final week when they can do that, and if they didn't do it, they might get the worst grade they expected, and that's important. Today, I am really delighted again to have a great speaker in our restoration seminar, again our last seminar, and we actually put the crown on this today, Ray Callaway from the University of Montana. And again, I need to tell a little story about that. Ray and I knew each other since 2008, and we have been collaborating on a fairly great number of projects. And he was kind enough to have me in his lab. Since that time, I visited him a couple of times. And yeah, I think I just can say thank you for many aspects for Ray. But today, he actually took this talk, and for this, I would like to introduce him for the publicum, as you might not know him. Ray Callaway is a professor at the University of Montana, Missoula. He has taught at UM since 1993 and is the recipient of numerous grants from the National Science Foundation and W. Mellon Foundation, USDA, the Murdoch Foundation, and the National Geographic Research Foundation, to name only a few, because there will be probably a whole page that we could say. He is a community ecologist with a primary focus on plants. To date, the Callaway lab at UM has graduated 10 PhD students. I think it's over then. That might be not accurate.
It's probably close.
Close to it.
11, maybe. I know.
Okay. And has welcomed visiting researchers from around the world, including Americas, Canada, Mexico, Chile, Europe, from Scotland, Hungary, France, Switzerland, Germany, Spain, from Asia, Turkey, China, the Republic of Georgia, India, and also Australia. So he has all these links all over the world, which also help him to be a great scientist. And he will tell us about his perspective of restoration and the rule of invasive plants. Thank you, Ray.
Thanks, Robert, very much. Appreciate that.
Thank you.
All right. Can you hear me in the back with this thing? I can't hear a thing coming through.
It's only a microphone that records.
Oh, I'm just wearing this for you.
Oh, yeah.
Oh, okay.
It's
I'm not sure, but this might be the first seminar in like 30 years as a professional that I've ever given like really on restoration ecology. I'm not a restoration ecologist. Like Robert said, I'm a community ecologist. I'm interested in what makes, natural systems kind of tick. But what I've tried to do, well, I've started off with a restoration slide, and what I'm trying to do is to take the research that I do on exotic invasives, which is probably 3/4 of the work that I do, and connect it in hopefully a meaningful way to restoration ecology, which is what you guys are interested in. And to do so, I did what any smart person would do that doesn't know anything about a subject. I Googled invasive weeds and exotic and restoration. And I came up with this actually really cool paper. It's published very recently by a woman with the last name Wallace and colleagues, in which they produced this cartoon, which we all know, but it kind of gives us a nice little picture of where to start. And so what they said is that, hey, look, in the system that's unrestored, we get the property that we like, whether it be natives on the ground, whether it be something about sort of the nitrogen cycle, is not very good. The property's low. We want to go up there where the property's high. So we do a bunch of stuff. We plant plants, or we dredge a channel, or we do things to try to bring that ecosystem property back. And we can get all these kinds of responses over time. And what I want to point out is that some of them are not so good. And I think around here, we could probably relate somewhat to that, because it's difficult to restore our grasslands to some degree. You can have really successful responses where everything's hunky-dory right away, and you can have failed responses, those light gray bars, or you can have really long time lags and finally get some success. And they argued in this particular paper that exotic, and again, we know this here, but that exotic invasive plants, or just exotic plants, played a pretty important role in the system that they studied in retarding the successful restoration. And what they looked at was forests like this. They're kiwis, they're from New Zealand, and they looked at their rainforests, and in particular rainforests that were being restored in what they call kind of urban forest lands like this, so kind of spotted throughout Auckland. And to make a relatively long story short, what they argued was that they did a bunch of correlations. How did canopy openness correlate with the amount of weeds? How did that correlate with tree basal area, epiphyte density? Those green boxes are what they want. That's their ecosystem property. But they argued or found that the presence of weeds, and again, this is just to get us started, we know this, that the presence of herbaceous exotic weeds had a very negative influence on the regeneration of tree density, and then ultimately probably epiphyte density. And so the whole point of their paper was to say, hey, we could do a lot better at restoration if we didn't have to deal with these exotic invasive plants, or exotic invasive species in general. And that's the whole angle I want to take today. And I do want to emphasize that probably they're working in a system that's relatively mildly invaded, in which the problem is relatively mild. In Robert's work around here, trying to restore things like tailings, are actually our grasslands, our intermountain grasslands, in my opinion, are much, much harder to restore in the context of exotic weeds. So there's some background, and what I'd like to do now is to focus on this, the why. And I don't know if I know this reason, but I'm going to take a particular direction and talk a little bit about why it may be so difficult to establish natives in the context of local exotic dominance. And to do that, you're going to have to be patient with me and let me be sort of a kind of an ecologist, evolutionary biologist first. And I'd like to take a step back and think for a few minutes about some of the most important ideas and directions in biology at all. How many of you guys have heard of this guy? He's kind of my hero. He's kind of a crazy Scottish guy that lived in the jungle and liked to shoot things and sell their carcasses and furs and his collections back to wealthy people in England. But as he did so, he was also a really good natural historian. And he made some observations that actually put him on the same level of Darwin. He was a contemporary of Darwin and probably came up with the idea of evolution and speciation right about the same time. And in fact, if I remember reading correctly, it was a letter from Wallace to Darwin that said, hey, I've been thinking about this evolution thing. That got Darwin cracking and actually made him get the origin of species out a little bit faster because he thought he was going to be scooped by Wallace. But what Wallace observed is central to biogeography. In fact, he's kind of considered the father of biogeography, and I'll explain what that means in just a minute. In other words, the distributions of plants and animals at the scale of the globe. And what he did is he worked in this area. This is where he had his little cabin and where he liked to collect things. And he made collections on these two different islands. And I'll give you a quote in just a minute, but he observed on these two different islands that one was just dominated by species that really had an Asian connection. And then you go over just a little straight and you find a place where species are dominated by, the flora and fauna are dominated by things that have an Australian connection. And from this he decided somehow these two groups of organisms had to be kept separate, and somehow the only way you could get at something like this is if this sort of isolation, geographic isolation, led to evolutionary processes or speciation. So his observations, and actually Darwin's observations, were biogeographic as well, just at a smaller scale. Here's a quote from him. In the archipelago, there are two distinct faunas rigidly delineated, which differ as much of those as those of South America and Africa, and more than Europe and North America. And we know now from doing real biogeographic studies that this is right on. And I'll show you a map in just a minute that'll illustrate that. So the bottom line is that we know now that plate tectonics is responsible for this. The continents have been zooming around the surface of the Earth for more than a few hundred million years old. And as evolution has progressed, and as groups of organisms have been separated, this is the foundation of evolutionary biology, we realize that geographic isolation is absolutely key to speciation and diversity on our planet. So this is the bedrock to thinking about evolutionary processes. And if we look at the distribution of plants in terms of floral kingdoms, we can see really key, we can see patterns that were described very well by Wallace. North America and Europe, at least the northern latitudes, have only been separated relatively recently. And they've been reconnected multiple times. You know, the New World tropics and the Old World tropics have been separated for 80 million years, and they don't share species. They share genera and families, but they don't share species. Up in the green part of that map, some species occur all the way around the globe. So these are now the evolutionary byproducts of 200 million years of the evolution of organisms and geographic isolation and speciation. So, in the last 400 years, we've not been isolated so well. And some people like to use the term Anthropocene. I don't really like it very much, but it's kind of hip. But we know now that those geographic barriers have been broken down like crazy by people for about 400 years. I mean, this is just a blink in sort of in evolutionary history. And we have, through various modes of transportation, distributed or redistributed pretty much every kind of taxa we can think of beyond its original range limits. And I mean, there are two perspectives we can take on this. You know, big deal. They have the same traits. They function just the same in the two different ranges. That maybe even adds to diversity in their non-native ranges. Or we can ask whether or not evolutionary history for organisms like these creates a situation in which they actually have bad impacts or negative impacts that we may not want, particularly in the context of restoration for you guys. I only work with one group of these organisms, plants. I mean, I'm interested in all aspects of ecology, but this is something I know a little bit about. And these are all examples of plant species that have been moved by people from one place to another place. And in their new range or their non-native range, they behave really differently in terms of their ecology. And that's what I'm calling an invasive. Other people will use the term differently, but I'm kind of restricting it to plants from exotic invasive to species that have been moved from one part of the world to another, and they seem to undergo sort of an ecological transformation. And my question is there evolutionary history part of this? I mean, can we compare their behavior in their two different ranges and find differences that has community level ecological implications? So this is where I want to go today. Do the properties of communities reflect geographical isolation? So in other words, geographical isolation we know is absolutely crucial to understanding evolution. Could it have community level ecological sort of implications as well? And that's actually a little harder to figure out. But this is a central part of where I'm going to go next. So, if you guys are interested in this kind of stuff, read this paper. This is written by kind of the god of studying exotic invasive species, Dan Simberloff at the University of Tennessee, and he kind of went after this question that I asked a little bit, or kind of what are exotic? Are they bad? Are they good? Are they neutral? And he wrote a paper with a bunch of people that studied the literature, and they came up with a kind of a rebuttal to another paper, which is basically, natives can act like invasive. Yeah, sure. I mean, lodgepole pine probably. So natives can be restless, but not very often. And it's almost always when they get disturbed. Some kind of type of disturbance is involved. And his point is that exotic species are many times, 10 times more likely to be considered problematic by humans, by us, there may be bias there, to be noxious or a problem. Okay, so he weighs in very strongly on the side, no, where you come from matters to some degree, evolution matters to some degree. This is just me browsing the internet. And I just want to point out, for whatever, there's maybe a lot of just human bias in this, but that pretty much any list of noxious species you want to find from any political entity anywhere, they're almost all exotic. Okay, maybe we just don't like exotic plants. Maybe this isn't fair. But Australia, for at least the whole country, the whole nation, every single noxious species that is listed there is exotic. They don't list any natives as noxious. Just take it for what it is. California, they're more tolerant. About 85% of the species they list as noxious are exotic. We're not tolerant at all. All of our noxious species, the species we don't like, are all exotic. They're all from somewhere else. So that sort of sets up a pattern. We do see a pattern, you know, and it doesn't take, you know, some ecologist, you know, to figure this out. We see patterns in nature in which exotics become really dominant and hard to deal with, at least when we want natives. So, can we find ecosystem/community properties that show biogeographical differences kind of in their function? And I want to start off with ecosystems. I don't know that much about ecosystems either, but this is really, really interesting. So this is kind of this is bedrock, right? Global annual net primary productivity. This, we know what does this. It's temperature and it's water, okay? And this has always been thought to be fixed. In other words, you can't change, you can't get this red to go any higher. You can't get that blue to go any higher. This is determined by the abiotic environment, a net primary productivity. That is sort of an ecological or ecosystem kind of law. Well, turns out we should be scratching our heads about this. This guy here, Liao, in 2008, did a meta-analysis in which he studied about 100 to 150 invasions. And what he found quite consistently, that ANPP in invaded areas compared to non-invaded areas went up, almost doubled. That's crazy. That's really weird. In other words, in a plant's non-native range, it's changing this fundamental ecological rule. And at the same time, nitrate went up significantly, or I should say 2 forms of inorganic nitrogen, and available nitrogen went up as well. So what that indicates is that these exotic invaders are doing something different from an ecosystem perspective. Don't know why. We still don't know why. But the pattern is really pretty strong, and this is across many studies. I had a student down here in the corner named Morgan McLeod who got interested in this and tackled 4 exotic invaders in intermountain grasslands around Missoula. And she started off by doing something, well, one of them here, leafy spurge. And she started off doing something relatively simple and straightforward. She just measured primary productivity and nitrate in the patches and out. It's correlation, not causation. And what she found was roughly what that meta-analysis showed. For cheatgrass, for Centaurea stevie or knapweed, or for leafy spurge, we saw about a doubling in productivity in their patches. Potentilla recta didn't do anything. And at the same time, just like Liao et al. Found, one form of available nitrogen about doubled as well. Again, we don't know why. She repeated this in a common garden, in other words, experimentally, and found roughly the same thing. And so this is Journal of Ecology 2016 if you're interested in looking in more detail. But basically she found the same pattern, and she also found something interesting is that the soil biota that are involved in nitrogen, making nitrogen available inorganically in the soil, skyrocketed. We don't know why this is. That should say 2016, I'm sorry. But she found that for all of these invasive species, she was getting something like 5 to 10 times more of these ammonia oxidizing bacteria. And why they should be promoted in a patch of invaders, no idea. But nevertheless, the pattern is a little bit ridiculous. And then finally, she found that annual above ground net primary productivity and its relationship with nitrate were different for exotic patches, the black dots, versus the native patches. So if you put all of this together, and actually a whole bunch of other studies, it does look like a lot of exotic invasives, I don't know about exotics in general, but exotic invasives do something differently from an ecosystem perspective, and we don't know why. We can talk about that afterwards, but there's a few cool hypotheses out there, but not really any evidence that I know of for any of them. I want to take one more step before I move from ecosystems to communities and talk about some unpublished work that Robert was very involved in, and maybe David, I don't know if you collected any of these data. There's the biogeographic pattern. I'm going to come back to the species for Solidago gigantea. This is one of our natives that has gone to Europe and has become a bad thing. And so this gave us the chance to compare these ecosystem properties in North America and Europe. And so the black bar is Europe, white bar is North America. Look at productivity at the top. What that really is a proportional change relative to non-Solidago around the patches. And in North America, the native range, it does what everybody else does. In Europe, it increases productivity by about 30%. Okay, what about nitrate? Well, in North America, there's at least a correlation towards less nitrate in Solidago patches, whereas in Europe, the invaded non-native range, we see the pattern that I showed you in the previous slides. So for whatever weird reason, there appears to be a biogeographical pattern. This species appears to be doing something from an ecosystem context that's different. And it would be cool to know if this really reflected something about its evolutionary history in a community context. And I'll come back to the species in a little bit and talk about communities. For you guys that are interested in restoration, there is an important aspect in this paper. Actually, you should all, if you haven't, you guys should read this paper. It's not just the presence of these exotic invasives that impedes restoration. It's often their legacy effects, and that's what these guys wrote about. So in other words, and through these ecosystem processes, the legacy effects could be strong. And if it is an increase in nitrate, a high nitrate generally favors fast-growing, invasive, exotic-type species over our relatively more stress-tolerant species in our grasslands. So anyway, I'll show this slide again in another context. So now I want to switch to communities, okay? And I want to talk about impact, and I want to talk about biogeographical comparisons of impact. And I'm only going to do this for a very few species and do it relatively quickly. But the idea here is I'm going to show you some, again, correlative measurements that indicate all of these species have been tested in this way, that the impact of these species is different in their non-native range than their native range. And that does suggest maybe that evolutionary history matters for community organization. And that's what pushes my buttons. So I want to talk about a species that honors Wallace's line. Eucalyptus is, you find it in Australia, lots and lots of species. And with a lot of people like Robert mentioned earlier, we measured the correlation between the canopy of this thing, which is thought to release litter. We think we have evidence that it does. That is inhibitory to other plants. We measured understory diversity. That's it. It's very simple. Species richness, native range, and several places where it has been introduced. It's not very invasive. It doesn't spread, but it does seem to have, as you can see, a pretty strong impact. Whoops, wrong way. So here's just for, I've not been here, but this is a picture of that same Eucalyptus globulus in the native range with a very lush, dense, species-rich understory. That's a picture you don't usually see in the non-native range. It looks more like that, typically, to my knowledge. Here's the impact on diversity or species richness. This is super conservative. If one species is present in the plot, then it gets counted. But what we see is that in Australia, yeah, diversity goes down under eucalyptus, but way more in the four different parts of its non-native range. The non-native range effects are double to triple what you see in the native range. So there's one example, and I'll come back to why this might be, but here's one example of a biogeographic difference correlation in terms of impact. Prosopis juliflora is another global invader. It's native to Venezuela and maybe parts of Mexico. It's a genetic mess, so it's a little hard to tell. But it's been introduced all over the world. So I was actually just looking at this species and working on this species last week in the United Arab Emirates. And there, I mean, they have hardly any species. It's a desert. But this thing does very well and has a pretty large impact on the species that are there. So basically, Did the same thing, it's just measured diversity under the canopy relative to outside, but did it in native and non-native ranges. So up above is what Prosopis juliflora does in its native range of Venezuela. The black is under the canopy, again, species richness. The white is in the open, and in its native range, it's a facilitator. You get much higher diversity under the canopy than out. It's a very arid area. This is a common pattern. But in two areas of its non-native range where we explored, and this is the same pattern was obvious in the United Arab Emirates. So the diversity goes down underneath the canopy. So it's a really strong difference in sort of the statistical interaction. So non-native range suppresses diversity, native range facilitates diversity. Again, it suggests that evolutionary history may matter in the context of how communities of organisms get assembled, and that has implications for restoration and what these kinds of species may do to kind of retard or limit restoration goals or successes. Last one I'm going to give you an example of, because I had to get Robert in here somehow. Solidago gigantea. This is the same species I showed you the ecosystem level effect of a minute ago. Solidago gigantea, again, North American, gone to Europe, causes trouble. Here's a picture. This was taken by you.
Hungary, yeah.
And that's impressive. And doesn't do this very. I mean, it can be pretty aggressive and big and dense in North America. In some places, particularly in the east, in Solidago canadensis, but it never lasts. And I've never seen anything quite like this. These are virtual monocultures. So what Robert did was he would only sample in Hungary, as you see. He would not cross the border to do any sampling. Well, in North America, I think maybe his non-native range, he was a little more expansive, and he went to 3 different states and basically did a pretty straightforward thing. 2 meter square quadrats, counted all the species, and counted the density of the stems of Solidago. And then correlated Solidago getting more dense with, again, species richness of the richness or the abundance, or the number, sorry, of other species. And again, there's a biogeographic pattern. Here's stem density of Solidago. Down there's a lot of it. Here, there's none of it. And there's species richness of everybody else. In North America, it's hard to get densities that they could find in Hungary. But in the patches that were dense, there's still the same number of species in those patches. Solidago is not suppressing everybody in its non-native range, but in its native range. In its non-native range of Hungary, there's a very strong decline in species richness associated with Solidago density. So again, this biogeographic pattern that I've been babbling on and on about. So these are patterns of community responses, and now probably have twice as many pictures as these. But these are all examples of species native to one part of the world, introduced to another part of the world. They seem to become bad players in their non-native ranges, and for which there's evidence of biogeographic differences and impacts. So there's quite a bit that has begun to develop. So, kind of 2 community scale components left to my talk, and then hopefully we'll have time for some discussion. But there's a lot of different hypotheses. There's way more than this. There's a zillion hypotheses for why exotic species often, go crazy. Underutilized niches by natives, they're adapted to humans, they hybridize or evolve in their non-native range, and there's some evidence for this, for all of these. But of course, the big dog is enemy release. You get away from things that control you in your native range, and you're free to kind of go crazy. And there is pretty good evidence for this. This is the basis for biological controls. We have some examples. Biological controls are specialist herbivore insects that will only attack, hopefully, the bad guy. And there are examples of biocontrols having some pretty big impacts. Most probably, by far, have not. So enemy release, yep, there's evidence that getting away from specialists, herbivores, helps, but not much. So that's where ecologists are really focused, but I'm going to focus somewhere else, and I'm only going to give results from one study. But there are many of these studies. I'm going to focus on the soil. And if you wanted my 2 cents, an unsubstantiated opinion, I would argue that the literature on enemy release from soil biota, whatever's in there, is probably the most consistent literature we have for any hypothesis on why exotic species sometimes go bad. And we don't know mechanistically very much about this. But I'm going to show you kind of one kind of big study from sort of a biogeographic perspective. But there have been others. This one is just relatively recent. We don't know the mechanism, like I said. We don't know if it's mycorrhizae. We don't know if it's like little mini arthropods, fungi, bacteria. There have been some isolations of fungal pathogens that seem to be, you know, the bad guys, but not many. So this study is focused, again, on species in Montana. These 5, they're all from Europe, where they all are relatively well behaved. Some of them are actually kind of uncommon there, but they all, I think most people would agree, somewhere in Montana, all of these species are pretty bad, have pretty strong impacts, and most of us Montanans don't like them very much. So, through these 2 people up at the top, the lead on this paper is John Marin. He's a colleague of mine at the University of Montana. The person that did all the work was over, not the little one, the older one. On the right is Lauren Waller. She's now a postdoc in New Zealand. She went to Europe. And that there's some really weird aspects of this study that are so cool. But she went to Europe and basically wandered around and found relatively native grasslands and collected soil. And then she took kind of broad sweeping vegetation sort of patterns. Who's around? Are any of those 5 species around in the grasslands where she collected soil? Came back to Montana, did the same thing, and then did what's called a plant soil feedback experiment. So you're going to have to be patient with me. I probably should have put a slide in. Plant soil feedback experiments, and again, if I'm not clear right now, this is a good time to stop me and tell me to do it again, is when you grow a plant in a soil for X amount of time. This just gets 2 months. And you let that plant species kind of condition the soil. And then you take it out and you use that soil, either a filtrate or that soil itself, either with sterile or non-sterile aliquots. And you say, what happens when I put another plant of the same species, expose that plant to the biota in that soil? And by far the most common thing you find in native soils is that if a plant is allowed to condition a soil's biota, that will be bad for the next of the same species. This was a discovery in the late 90s. Fantastic stuff. It's all done in the greenhouse, so it's hard to interpret in the big natural world. But hopefully that's clear. It's what a plant does to a soil, it's biota, that in turn affects the next generation of the same plant, same species. So here's our 1, 2, 3, 4, 5, 6. Did I have 6 pictures? I don't even remember. 6 bad guys, and their feedbacks in either soil from Europe, which is the native range, or soil from North America, Montana, which is the introduced range. We got no significant feedbacks in introduced or non-native ranges. This is very, very common. I'll tell you what's weird about this study in just a second. We got 4 out of 5. I mean, that's an incredibly strong feedback. That means Centaurea stevie, and we've done this now multiple times, can barely grow in conditioned soil, okay, with the biota that occurs there. The same with Linum and Potentilla recta and Euphorbia esula, spurge. So we saw a lot of really strong negative feedbacks, native range soil, nothing from non-native range soil. This is common. This has been explored in meta-analysis. This has been shown a lot. I really do think that the regulation by soil biota in the native range of many, many species is really very important and something that a lot of exotics escape. And again, perhaps more important than the effects of exotics on ecosystem processes, such as nitrogen cycling, this could take some time to go away in terms of restoration. So this is another potential area in which legacy effects could develop. So these are those hypotheses for why exotic plants might be successful again and again. Like I said before, there are many more. It's important, though, to note, one, that these are not mutually exclusive. These can operate at the same time. They may be different in terms of their relative importance. Again, I'd put my money on soil biota for being the most important. But I want to wrap things up by going back to Eucalyptus the Eucalyptus species that I was showing you a few minutes ago, and show you evidence, just one slide, even though now we have, I don't know, dozens and dozens of slides along these lines, in which the biochemistry of the plant may play a role in its invasive process. This could also maybe have legacy kinds of effects. It's wrapped around the novel weapons hypothesis, and this is the idea that plants are little chemical factories. And even within one species, you can have different ecotypes that are completely different in terms of their overall biochemical signature. Like thyme, what we like to put on our food, is native to Europe, and its chemotypes across the landscape are absolutely fascinating. So plants, again, first are little species-specific chemical factories, and this idea is that maybe when you bring one of these little species-specific chemical factories into a new range, whether it's through the chemical effects on other plants, chemical effects on herbivores, on soil biota, all of this stuff, maybe there's an advantage once in a while to bringing some novel biochemical signature to a new place that nobody's had a chance to see or evolve to. And we tested this. In my lab, we've tested this for a number of different things, but this is probably one of the best results we've ever gotten. It's for Eucalyptus, the plant that I showed you had those biogeographical differences in impact. Morgan Luce did this as well as when she was a lab tech instead of a master's student. And all we did was run water through naturally collected litter, make this out of it. Can you see that? It's just natural litter under the trees. Pick it up, put it in run water through it, and we get this. It's like a black tea. We applied that to seeds of species from the different ranges. So these are Australian seeds. A whole bunch of them did germinate. So this is a pretty low replication. And then seeds of plants that are native to California, native to Chile, and native to India. Those are some of the ranges we looked at, the understory effects. And in the native range, the overall effect is kind of a wash. Everything from a couple of negative effects, a couple of positive effects, 2 neutral effects. So in other words, we got everything in the native range in terms of the impact of the litter. Every single species we tested from the non-native range was really strongly suppressed in terms of the change in root growth. It was really strongly suppressed by eucalyptus litter. My brother's a high school teacher. He's had students play around with this. I've got him seeds as in sort of their high school science projects and basically found really similar things. So what's cool about this and studies like it is it does indicate that biochemistry, just in a really simple way, this is just litter effects on other plants, that biochemistry may be a component of this evolutionary history that indicates biogeography and where you come from in terms of being invasive may matter. So in terms of that pattern, in terms of a biochemical signature that seems to have differences on species native to different parts of the world, has been shown for a lot of plants. Again, now it's more than this, but this is a collection of species that people have. This now is not all from my lab, but that people have experimented with and said, yeah, it looks like there's biochemical aspects of these plants that have different effects in their native and non-native range. So last slide, and then maybe we can talk about this for a little bit. I do think, and I've just cherry-picked some of these biogeographical patterns, but I do think there are some properties of communities that I've shown you and ecosystems that I've shown you that appear to reflect, geographic isolation. So is one implications for restoration, man, if evolution really matters, it isn't just like, leafy spurge just acts like, your typical forb, Montana forb. There may be a reason why it's so hard to get out of your plots and restore them, because you're fighting against sort of an evolutionary disadvantage. Leafy spurge, for any of the reasons I've talked about, might be freed up to do stuff to natives that makes it very hard to restore. It may take the development of more and more resistant genotypes of natives from North America to deal with something like this. In terms of biology or ecology, it does suggest that geographic isolation is not just foundational to evolution, like where I started, but it's also really very important to understanding how communities get put together, and then also how they fall apart. So I'm done. I assume we're done at 10 till? I was just hoping I was trying to hustle so I could leave some time for questions, but I thanks very much, and I'll try to answer the questions.
So in one of these fields where you've got stuff growing all over the place, has anybody gone in and maybe either randomly or maybe not randomly, harvested plants around in the field, one here, one there, and then watched to see if that plant robustly reappeared in the spot where it was harvested? In other words, you took the root and everything out, so it's got to reseed itself, I guess, in that spot.
In the context of the plant soil feedbacks, or just in general?
Well, just in general, because you described that experiment where they came in and they put the same plant in a place where the
And this was in pots, so it could be controlled.
yeah, but
Yeah.
what if you did it?
Got it.
What if you did it in a less controlled environment?
Yeah,
What
it's
kind of field would that be?
there's a guy at Utah State, Andrew Kulmatisky, who has developed some protocols for exploring plant soil feedbacks in nature, finds them. Plant soil feedback patterns often correlate, but it can not always, often correlate with the abundance of particular species in natural systems. And there's also some evidence that there is a dynamic quality to the spatial arrangement of plants, just like you asked, that might, don't know what it's related to, but it could be, at least it correlates with this feedback thing. So in other words, where you have species A in an old field in eastern North America, 3 years later you're going to have species B. But if there is a study that connects that more explicitly to plant soil feedbacks, I don't know it. So, Beth.
So right on, different question. Can you go back to one of the graphs where you showed, it was before that.
Well, there's
It had to do with one of the plants, and unfortunately I don't know any of the plant things. It was.
But what was the topic?
It was, keep on going back. It had to do with the number of things, number of things growing in the open and growing in the understory. Is that right? Is that one?
That's that one. How do I get this back up here? There we go.
So I'm looking there and I'm seeing in the subcanopy, there are more than out in the open, but the number of species is like 7.
Yeah.
If I go down to the non-native species, in India, in the first non-native species, in the subcanopy, there's actually 8 species there.
Yeah.
It's more than 7.
Yeah.
Not a lot more than 7, but it's more than 7. And so I'm a bit curious about, and the numbers were significant. You didn't show very many cases.
Say again?
I mean, the numbers, me picking on the number 7 being almost the same as the number 8 may not be as significant.
No, probably it's not. This is a desert, so in other words, it has to be calibrated by what you find naturally. You got a species pool, and you have to start with that. And what matters here is the effect, the relative change. In other words, you can't do anything about what the number of species that are in the species pool at a particular place.
Right, but there are obviously at least 7 species in the species pool, because if there weren't 7 species in the species pool, they couldn't be under the subcanopy.
Correct.
Are they all invasive? No, they're not invasive.
These are natives.
Right?
These are natives.
And then in the non-native ranges in Hawaii,
2.
Right, 2, or 3 is 2.25 or something, and then 8 out in the open. So, but what happens there? What happens underneath these plants where they don't belong?
I'm not sure what you're asking, but what I can do is present data that show in one place there's a facilitative effect and in other places there's an inhibitory effect. We did experiments with this litter as well, but we didn't do it, so I didn't show it because we didn't do it biogeographically. We only did it to No, this is Prosopis juliflora. We only did it, I believe, to species native to India. And what we did is we compared the litter, assuming this is what you're asking, we compared the effect of the litter of the invasive prosopis to the effects of the litter of a native prosopis, Prosopis cineraria. And we got huge mortality from the litter of Prosopis juliflora, which is full of a particular...
Just the invasive.
The invasive litter killed Indian natives to a much higher extent than the litter from the native Prosopis. So it's not a good biogeographic comparison like I wanted to show you guys, but it does indicate that the litter can be nasty, much like the litter leachates from Eucalyptus. In this case, we just applied the litter.
Right, but in those non-native ranges, like take Hawaii, how many species would normally be in some, acre, square, hectare, whatever units are? Is 8?
This is a comparative sample under and out. That's it.
Right, okay, so, but then, and it's not under something else, or if I looked under...
It's open. It's open. It's outside of the canopy.
Outside the canopy, but
Yeah.
I had a different canopy.
We've done that. We've done that as well.
Then, what's it like under a native canopy in Hawaii compared to what's it
Yeah.
like?
In other, you're asking.
No scale.
You're correct. You don't have any scale. In this particular, we've done canopy comparisons as well, but it depends on the species and the system. And there's evidence for, not this tree, but for other invasive trees, that canopy effects are very different than the canopy effects of natives. Bill.
You're showing us examples from grasslands. Do these trends hold with the invasive grasses?
Invasive grasses.
Can you see these kind? Have you experimented with it? Grasses?
Me myself with cheatgrass, and I don't know if I mean across the board plants row feedbacks.
Oh, well, I'm thinking about the European pasture grasses. Dominate so much in western Montana and
Yeah.
North America. What do we know about those? Does it fit this pattern at all?
Yeah, the best examples that I can think of would be things like Bromus dinermis, thank you, which you see often roadsides moving out. It's not exactly like knapweed kind of parachuting into the middle of nowhere and seeming to do bad things. You've got Agropyron cristatum, more invasive north of here in Canada, but I don't know of examples. Johnson grass, however, which is more invasive in the south. It's a C4, so it's more invasive in the southeast. It's both an agweed and an exotic invader. Some of these principles have been tested for sorghum halopense, for example, the kind of allelopathic effect. And Johnson grass is probably one of the better examples anywhere of kind of knowing from chemical to the field, kind of how it chemically affects other species. So, but I don't know about here.
Yes, I wasn't thinking about the European pasture grasses that are not noxious, or brought here to improve pasture and things like that.
You were or were not?
That's what I was wondering about, whether the European pasture grasses that are not really noxious grasses.
Oh, I see.
They're feeding our cattle and whatever.
Yeah, I mean, I've only seen most of these tested with species that are at least thought to be invasive. Yeah, to my knowledge.
Ray, you show a slide the global distribution of ANPP.
Yeah.
And this data is from remote sensing data, or is from kind of a network?
Say again?
The map you're
Yeah.
showing the ANPP.
Yeah.
It is from remote sensing data, or it's just
I'm sure it's
network?
I'm sure it's a combination. I just picked that to introduce the idea of ANPP and the idea that this is thought to be tightly controlled around the globe by temperature and by water, and that we see invaders kind of breaking rules that we expect to be pretty solid in ecology. That's all. I don't know anything about the map or anything.
Yeah, from normalization, we can produce a time series.
Yeah.
Spring all the time to the fall.
Okay.
So you see the maximum productivity map. So probably you will see the maximum or the average.
No. No, what we're seeing is that exotic invasive species change ANPP on a very local level. And that's the whole point, and that's what's weird. And again, that's what's weird if it's actually true across maybe at a larger scale, or if it's sustained over decades and decades and decades, and we don't know that. So, sorry, you've been raising your hand, and I'm not.
So from a definition standpoint, what's the difference between a non-native and an invasive?
Well, you're going to get. I'll give you my answer, and I tried to lay that out at the beginning because people will argue about the terminology, and so I tried to be clear up front about how I'm using it.
Okay.
So, I like the term exotic invasive. Okay, and the exotic, you're not from North America. You're from Europe. You're from somewhere else in a biogeographical perspective. Invasive, there's some evidence out there, either just with your eyeballs or with actual quantitative measurements, that your ecology changes. You either become far more abundant, you have higher impact, your spread is more rapid, you make bigger plants. All of those things have been shown over and over again, comparing species in their two ranges. So then you can say, okay, it's invasive, in other words, but that is actually a judgment call. Some people would argue that that species A is it's not really eucalyptus. Some people argue, that's a bad invasive. Other people, no, that's not invasive at all. And so that gets a little bit sketchy. I think the species I focused on, people would agree that they really behave differently in their two different ranges.
A long time ago, Clements and Weaver used to argue, about whether there were plant communities or whether there were just species that just would just adapt and move.
Yeah, Gleason would roll over in his grave. This is very Clementsian, and this isn't Gleasonian at all. But this, Clements would argue that, hey, maybe there are meaningful evolutionary things in communities,
right.
But Gleason argued no. We know now that Gleason's wrong. Gleason would argue that no, these are population phenomena, and that's it. They just happen to occur in the same place. We know now that's no longer really, even though that's what's taught in all the ecology classes. Modern research indicates that that's really not the case.
May I ask? I don't know, kind of the last one or before last one. So in Butte, I kind of just try to get the Butte perspective out now, because we need to do restoration, and you pointed out it's really crazy to do here. And the problem that we face here is one of the Eurasian grasses that we need to fight, because there have been reclamation here. And I picked out, that was really great to see that biogeographical perspective, that one place you have this much biomass growing, and on the other side of the globe you can have this much biomass growing. So here in that part of the world, they might be, where actually we are kind of restoring or reclaiming things, might have been a shortgrass prairie. And basically with these grasses, EPA actually asks now us to put back at least 60% cover, right? That's our thing that we need to achieve. But if we go out to the shortgrass prairie and we sample, that's not the case we actually get. So they kind of forcing us to do that. And one way to do it is very successfully to do it with a no-native grass, with crested wheatgrass. So I kind of just put that up in the air that, what is your idea about that? I mean, we need to find ways about fighting and making the grasslands more diverse, but it's a fight that we actually we're going to find.
But I mean, there is a judgment call here. So for like the species Bev was curious about, Prosopis juliflora, that has provided a wood resource in very, very poor countries, sub-Saharan countries, that would make it really quite a negative thing to go in and say, this is invasive, we got to get rid of it. It's got costs. it comes at costs, but there are also benefits. And I think I don't know quite how to make that evaluation. I personally would rather see every effort made to restoring whatever system you're working on to something that resembles a native system. If indeed that means the whole mountain is going to slide down onto Butte, well then use an exotic. You just get, don't let that happen. But that's always going to be a tension.
Yeah.
And you got to know the costs. And a lot of the grasses now I kind of see where. Sorry, I kind of misunderstood. A lot of those grasses are not very invasive. The most invasive species probably in all of Western North America, I think by far, is cheatgrass. That dwarfs the stuff I've been talking about in terms of impact. I know we know nothing about what to do with it. But a lot of these restoration-related grasses, you're not restoring. It's not restoration. You're reclaiming. You're changing the environment there so you can get something that you want out of it, like not run off into the street. But you're not restoring it to, a native grassland.
And that's what basically EPA tells, in a way, that was just the beginning. So we actually now have a stabilized area here, so the migrants is not sliding.
Maybe.
But now we actually need to go towards restoration, which, based on reference sites, models, we just try to enhance that crested wheat grass. And we actually are facing all the problems. Mark, one of our grad students, is actually figuring out all the bad news about crested wheatgrass.
Crested wheatgrass, yeah.
All what you show here.
Yeah. I'd be interested to hear more about that. Yeah.
Okay.
Yeah.
I don't know. Do you have any more questions? Maybe Mark.
Just a quick one. So you did the leachate experiment on
Litter.
the eucalyptus litter, and is there a way after to possibly isolate the chemical or chemicals that it's actually doing that dirty work?
Yeah. And it's been done. For example, Johnson grass is a great example of sorghum is a really well-studied and understood chemical, how it works, mode of action on photosystem 2, how it's produced, where it comes from, root hairs, and so on. It's just really hard to do. And I am no biochemist. I got a C in biochemistry, and so I don't do So anyway, but there are people that are very interested in that. And if you wanted to really dive deep into some of the, kind of allelopathy, anti-herbivore, or defense chemical stuff, holy moly, there's just stacks and stacks of fascinating stuff to read. So people have looked at a lot of chemicals in eucalyptus litter. I don't know which one or which ones play a role.
I guess along the lines of allelopathy, do you see co-invasion or something maybe where you have
That's a great question.
And then the understory has plants that co-evolve with it to
I don't know how to take it quite that far, but you're talking about something that, actually Dan Simberloff, the guy that I mentioned a while back, has coined invasional meltdown. And so there is evidence for an invader coming along, changing the environment, and other invaders like this. And actually I've been exploring the impacts of multiple, the biodiversity of invaders on natives. And what we are finding non-additive effects, small, but that 4 different species can have a greater relative impact than 4 individuals of the same species in some cases. So I don't know about the evolutionary thing. That's a tough one to get at. But in terms of this invasional meltdown, that's a great question. Well, thanks. You guys stayed later. Thank you very much.